Photosynthesis, John Ingenhousz this identified that the plants can produce oxygen when the presence of light. When the presence of light. Mainly the green plants. Green plants can perform the photosynthesis, or it can liberate oxygen when the presence of light. Which plants? Green plants. So, in the presence of light energy. Okay. So, he performed this experiment, that is a Hydrilla experiment. First of all, you want to know the plant. What is the plant? Okay. Hydrilla plant. Hydrilla plant is a aquatic plant. It has a dense leaves, very small, tiny leaves. Right? That's the aquatic. It's submerged aquatic plant and this one. And the నీళ్ళ లోపల బతుకుతూ ఉంటుంది. And it can perform more photosynthesis. Why? Because it has a abundant chloroplast in it. Okay, so it will look like a green, very green. Okay, so that's why, and also it can easily liberate oxygen. Why? Because it can easily visible it. So sometimes you can observe from the water, we can observe some bubbles are coming from the water body, from the inside to outside. Okay, so that's because of there is a presence of plants. The plants releases the bubbles. That bubble contains which gas we don't know, but we will know with the help of this experiment. So we can use Hydrilla plant as well as we can also use Elodia plant. Elodia plant. So first of all, our experiment aim to prove that oxygen is released during photosynthesis or in the presence of light. Aim is to prove that oxygen is released in the presence of light or in photosynthesis. So next, requirements. Required objects is a test tube, funnel, beaker, splinter. Okay. So next, chemicals. We are only using one chemical, that's the water. Okay. So materials we can require, that is the Hydrilla plant twigs. Hydrilla plant twigs we need here. Okay. So procedure. First of all, if you see the procedure, a twig of Hydrilla are taken and inserted into the funnel. Funnel, it is a funnel. It is a funnel. We will collect the twigs and we will insert into the funnel like this. What we are arranging in the experiment, that also. Okay. So thoroughly we can insert that Hydrilla twigs inside of the mouth of the funnel. Okay, from wider mouth to narrow side we can arrange that. Okay, next the funnel along with the Hydrilla twigs is placed in a beaker filled with water. We will already take beaker with water, and this total arrangement will keep it in the water. Okay, so next one, after test tube. So we will take a test tube also. We can write here test tube. Okay, so test tube also we can take some amount of water in it, and we will use our thumb, because we can use our thumb and gently keep it on the mouth of the funnel. That the test tube will be kept on the funnel mouth. Okay, funnel's mouth. So that's it. So we will invert that one. Okay, we will take a test tube and place our thumb on the mouth of the test tube, and we will invert it and we will keep it on the mouth of the funnel. Next one. So next, ensure that the level of water in beaker is above the level of stem of the inverted funnel. So inverted funnel, if you observe, water is funnel kante paipuga. So we take it above the funnel beaker, and also we will take water in the test tubes, that also in level, the water level of the test tube is more than that of beaker's water level. Okay, so that's it. Next one. Ensure that the level of water in the beaker is above the level of the stem of the inverted funnel. So it is present. Okay, we have taken like that. So next, the entire step is kept in the sunlight nearly 2 to 3 hours. So now total arrangement will keep it in the sunlight 2 to 3 hours. Okay. After that, the level of water is observed in the test tube. So level of water will be tested. Why? Because we can observe we can observe when you keep that hydrilla plant or total apparatus in the sunlight. So automatically after 2 to 3 hours we can observe little water bubbles coming from the hydrilla plants. That water bubbles will be collected in the side of in the test tube. In the test tube. So after collecting the air or gas of the hydrilla released gas into the test tube. So after getting it, so we will separate that test tube with the help of our thumb, and we will keep some burning matchstick on the mouth of the gently you can leave your thumb from the mouth of the test tube, and we will place a burning candle or burning matchstick in the mouth of the test tube. It shows that the flame will get bright. It will flame is a brightly it can flame. Okay, it can burn like brightly. Okay, that's the observation. So after some time, small gas bubbles come out of the leaves of the hydrilla. The gas is tested with a glowing splinter or matchstick. Also, we can use here. Okay, so it means the bright to the flame because of presence of oxygen in the test tube. So that oxygen was prepared or produced in the hydrilla plants in the process of photosynthesis. Are you understand? So this is on the result. This so the result is in photosynthesis, hydrilla plant can release us oxygen.
Yes.
We can observe, we can prove with the help of this small experiment. Okay, like that, every plant can perform photosynthesis and it will collect carbon dioxide, water, and in the presence of sunlight and chlorophyll, it definitely releases oxygen to the outer environment. That's why we called it as a universal oxygen providers. Simply we can say, and also it called as a universal food providers because of every organism depends upon directly or indirectly on the production of food of the plants. Are you understand?
Yes, sir.
Up to here?
Yes, sir.
Any doubts?
No, sir.
So this is the experiment. A, objects, chemicals, materials, procedure, and finally your observation, and we will get some result.